In this video, I will explain the process of a skeletal muscle contraction. In our bodies, we have three different types of muscles. The skeletal or striated muscles are the muscles we can actively control, like for example the biceps and triceps in our arm. Secondly, there are smooth muscles, which we don't actively control, like the muscles involved in the processes related to digestion. Lastly, there are cardiac muscles. Cardiac muscles look like striated muscles, however, they are only found in the heart and contain intercalated discs which allow the electrical signal to pass through the muscle, causing it to contract. Inside each muscle are bundles of fascicles. The ends of the fascicles are attached to tendons which connect the muscle to a bone. Inside of each individual fascicle are bundles of muscle fibers. The muscle fibers consist of many motor units. Each motor unit has an axon terminal from a motor neuron. Inside each axon are vesicles with a neurotransmitter called acetylcholine. Inside the muscle fibers are bundles of myofibrils, which have multiple nuclei and many mitochondria surrounding them, as ATP is a vital part in a muscle contraction. Inside the myofibrils are the sarcomeres. Sarcomeres contain myosin and actin molecules. Surrounding the myosin and actin are T-tubules, which lie in the membrane of the sarcomeres and also play a vital role in the muscle contraction. The T-tubules consist of the sarcoplasmic reticulum. Part of the sarcoplasmic reticulum is a terminal cisternae, in which calcium ions lie. These calcium ions can only pass through voltage-gated calcium channels in the T-tubule. However, these channels need to be activated first. This is where the motor neuron comes into play again. When an action potential is passed down the neuron, the vesicles, which contain the acetylcholine, release the neurotransmitter. The acetylcholine molecules then bind to channels in the sarcolemma, the membrane of the sarcomere. This causes the channels to open and allows sodium ions to enter the sarcomere, creating an action potential. This action potential then passes along the sarcolemma and into the T-tubules to the voltage-gated calcium channels. These then open, releasing the calcium ions into the sarcomere. Why is this important? Well, to understand this, first you have to understand the roles of myosin and actin. Here, actin is in blue and myosin in red. The black stripes in the middle are a supporting protein for the myosin filaments and at the same time represent the dark band of the sarcomere. The myosin filament is the thicker of the two, and it contains many heads. The heads are always in pairs as the structure of one molecule is a double helix. The myosin heads have two binding sites, one for ATP and one for actin. Without the calcium ions, the myosin heads, which always want to bind to actin, are stopped by a contractile regulatory protein called tropomyosin, which covers the binding site on the actin. When calcium ions are present, they bind to another contractile regulatory protein called troponin. This causes the tropomyosin to uncover the actin binding sites. The myosin heads now can, by hydrolyzing an ATP, bind to the actin. The myosin heads then pull the actin towards the center of the sarcomere, towards the M line. This results in tension on the actin, which leads to a reduction in the length of the sarcomere. To illustrate this, here are two sarcomeres. The top one is relaxed, so without the calcium ions. The second one had the calcium ions released, and so, because of the myosin, is pulling the actin towards the center of the sarcomere. The length is therefore reduced. When a strain of sarcomeres do this, it means that the muscle is contracting. And that was the contraction of the skeletal muscle.